our Dendrites workshop, Dendrites Necessary for Cortical Computation. I think we have a superb lineup of speakers and questions that we can address. Our Dendrites. And we will give 20 minutes time for each speaker and then move to a 5-minute question. So you should see in your YouTube a top chat or live chat window, and you can please, if you have questions, put them in that live chat and they will get conveyed to us, and then we'll ask them as a moderator, since we cannot have everybody in the audience come live to ask questions, but we very much appreciate questions. So let me give a very brief introduction. So dendrites, of course, are not a new phenomenon. So we have studied dendrites for a while now, and even I think with the foundational work of Will Rawl in the late 50s and 60s, we got to understand theoretically how they operate, how they conduct electrical currents, and can be viewed as cables, basically signaling from the distal ends towards the cell body with some sort of passive decay. And that is principally understood, the physics of that are principally understood ever since Will Rawl's work. Then for a few decades, work in physiology and modeling has shown a lot of very interesting active properties of dendrites, things like dendritic spikes or plateau potentials. And that has culminated even already in 1992 in a book, which is still interesting to read, called Dendrites. And then, of course, we also want to know, what can you do with these active properties? Are these dendritic nonlinearities functionally important? And models and theories have been formed about that now also for several decades. And for instance, we have a book called Single Neuron Computation, which is also featuring a lot of dendritic properties. Here you actually see depicted on the right a Purkinje cell, which we will not get a chance to talk about today, but is a brief tag for me to mention that not only cortical neurons have active dendrites, but that active dendrites in different brain regions may actually do different jobs. So that is a level of complexity we do not have time today to go into, but should be clear. So dendrites matter, and dendrites have been studied and are fairly well understood in their biophysical principles for some time now. And the field of neural networks also has been progressing now for several decades in complexity and achievements. Interestingly, though, those what we call artificial neural networks almost without fail will just have simple integrate and fire nodes and will not employ any dendritic properties. So one of the questions we have today is, why is that? Are we failing to convince folks who do computation in networks that dendrites are necessary? Or are we still lacking the requisite understanding to basically make neural networks operate at an even higher plane than they currently do? I wanted to point out here, and this is just a random sample of 2 publications I found, that even as late as 2021, when we discuss not only neural networks in general, but neural networks when they are applied to understanding the brain. For instance, deep learning neural networks have been shown to map in their visual field properties in some sense pretty well onto our different hierarchies in the visual system in the mammalian brain. But even in this overview article of how we understand to map deep neural networks onto brain function, there's not a single mention of the word dendrite. So even when we apply neural networks to biological problems, do we typically not think about dendrites? So that is the question: is why, and can this workshop sort of be a part of a movement that says, wait a moment, maybe we can do better? And speaking about doing better, are there ideas that guide us of what maybe is missing in artificial neural networks, and what do we want to actually achieve with dendrites? So here's an overview article from Matthew Larkin, who is a well-known researcher on dendrites and sort of has forged many ideas in the field. And here's a specific idea which has to do with dendrites, and you see sort of here the apical dendrite of a cortical neuron, and you see that the input there, the fibers, and this is really a large number of the synapses and the inputs the neuron gets are up here in the tuft. that a lot of the fibers that coming into the tuft are associative fibers, are fibers that provide feedback from higher brain areas. There are also fibers from higher order thalamic nuclei that impinge onto the upper layers of cortex. So one idea is that we have some top-down processing here, that we can shape our signal processing based on our previous experience, based on expectations, based on an internal model. And that maybe it is this top-down guidance of neural processing that dendrites are essential for. And I think one can fairly point out that this top-down sort of cognitive guidance is certainly something lacking in even the most sophisticated deep learning networks at this point. So, so this is one of the teasers, is can we support that? That is what cortical dendrites might do. So today we have a really expert panel, speakers who have worked on dendrites for a long time and sort of do state-of-the-art models in computational property determination. We'll start with Dr. Arnd Roth from University College London, who gives us sort of a biological principles of dendritic processing and cortical neurons. Then Dr. Brent Doiron from the University of Chicago will talk about specific inhibitory plasticity and assembly formation. Dr. Bartlett Mel from USC in California will talk about how to optimize a dendrite for binary subunit pooling. Dr. Yiota Poirazi from Crete will talk about dendritic solutions to logical operations in models of human neurons. Dr. Andreas Stöckel from University of Waterloo will talk about passive dendrites as a computational resource in the neural engineering framework. And finally, at sort of the highest level, Dr. Xiao-Jing Wang from New York University will talk about dendritic gating in the whole neocortex. And importantly, we will have time for a 20-minute panel discussion that will be hosted by Michael Häuser and myself. And we'll try to sort of ask cross-cutting questions and challenges and see whether we can extract a new state-of-the-art knowledge about dendritic function from these presentations. And again, please at all times use the live chat to ask questions. We welcome questions. I will get a copy of those, and then we'll have 5 minutes after each presentation and ask questions for that presentation in those 5 minutes, and then we will have additional ability to ask your questions also in the panel discussion in those last 20 minutes. And then without further ado, I would like to hand on the screen to Arnd Roth.
Thank you. Let me share my screen. So today I'll tell you about work that's been going on in our lab, Michael Häuser's lab in London, in the last few years, modeling responses that we can observe in vivo when we patch dendrites or when we image dendrites. This is all motivated by, as Dieter introduced, the wide range of dendritic anatomy and function that we've observed, and we've always been filled with wonder what the different structures and the different combinations of ion channels in different types of neurons might mean for the way in which they process input to generate output, which is, one way in which we like to formalize or describe the problem. We want to find the algorithm or the simple model that replaces the big question mark here, given a certain set of inputs, which of course we have to choose based on what we think will happen in vivo, what kind of inputs neurons will receive under different conditions in vivo. How should they be analyzed and treated by the neuron to decide when it should produce output spikes? And we like to think in a complementary fashion of this problem also as what are the different electrical or chemical functional compartments in neurons? Should we consider a single spine as one compartment, certainly for chemical signaling, for example, for the sinus specificity of LTP, but electrically? Should we consider a group of spines on a short branch? Should we look at the entire branch or the entire subtree? And depending on the input conditions and the mechanisms we study, the answers might be specific and different for those conditions. The study I'm going to talk about was really motivated by beautiful experimental work in the lab by Spencer Smith and Ikuko Smith, who heroically patched dendritic locations in layer 2-3 pyramidal neurons while the animal was either anesthetized or awake and was watching a video screen. And this is in visual cortex, so we get visual responses. We get cells that are tuned to a preferred orientation. This is with symmetric recording. This is what you would expect to see. And now what happens if you have, by chance, hit a dendrite is that weird stuff shows up, little bursts of events, and also events that are single ones that look like they are backpropagating exponentials. But in this paper, there is lots of evidence suggesting that not all of these events recorded in the dendrites can be originating at the soma because the firing rates would be just too high. And these events, similarly to the somatic output, have a tuning. Now, if you look more closely, we see that these events are not stereotypical. They vary in shape. So we can see there's the high rates. We can see the amplitude varies. So they are not likely to be all backpropagating unless backpropagation is very modulated by in vivo activity. And we also have different time courses of the spikes, suggesting that different ionic mechanisms might contribute. So candidates for this include, say, voltage-gated calcium channels, for example, voltage-gated sodium channels. But of course, mostly we think voltage-dependent NMDA receptors. So coming from these experimental findings, we were wondering under what conditions we should be observing such signals if we try to make a model. Sorry, I want to add this. This is a very recent paper from the UST lab showing with voltage-sensitive dyes that you can also see events that involve multiple spines or parts of a branch. And so the advantage here is that we have a fairly linear readout of even subthreshold voltage, unlike what you have with calcium imaging. So this is a paper you may want to look at. It just come out. The work I'm going to present is by a gifted PhD student from the lab, Lea Götz, and she addressed the following questions. If we run a model of a layer 2-3 pyramidal cell that is part of a circuit and receives input generated by the rest of the circuit, what determines whether this neuron will spike and at what output rate, and whether it will be a tuned output? Which synaptic input patterns can engage or can create dendritic events that we may have observed with the dendritic patch clamp recordings in vivo? And finally, if you compose those two questions, if you start from a single synapse, looking at dendritic events it may trigger, what does this mean for the somatic exponential output of the neuron? And so our approach has been to bring various tools together. One is we have to have a realistic model of the postsynaptic neuron, its morphology, its somatic, axonal, and dendritic excitability. And we have to optimize that based on available data. Next, we want to engage this with what we hope to be a realistic model of the synaptic input patterns it receives during spontaneous activity or visual stimulation. And in that regard, we took note of recent work from our colleagues in London who first did 2-photon imaging of visual responses of neurons and then made brain slices from the same mice, registering the same neurons. So they knew when they patched neurons in the brain slice what the response properties of these neurons had been in vivo. And what they find is that, as was well known, there is a log normal distribution of synaptic weights, of the weights in terms of somatic EPSP amplitude that you get when you activate the presynaptic neuron and record the postsynaptic neuron. And they were then able to relate whether a synapse was strong or weak to how similar their responses were in vivo, or how similar their tuning was to visual features. And the key finding is that similarity in response selects for, but not exclusively, strong synapses. So we built that in. In our model, we have neurons that have a similar visual tuning, preferentially, but not exclusively connected by the stronger synapses. And when we do that, we can indeed get behaviors that look similar to what Spencer and Ikuko saw in vivo. We get tuning at the soma, and we get these weird high-frequency, variously shaped events in the dendrite that are also tuned. And if you now want to say which is which here, which is the experiment and which is the model, it is, well, not so obvious. So hint, the experiment is on the right. What next do? We want to study what's really happening in the dendrites. We just want to use the fact that the model gives us access to all of these variables, like not just the membrane potential, but also the activation state of all the channels, the current through sodium channels, for example, or through NMDA receptors. Sorry, what's happening here? So we can benefit from this full, range of information that is not, of course, accessible to the experiment. But then, of course, it's a model, so it's only as good as the quality of the model in terms of representing reality is. But that problem being there all the time, we now go ahead and define detectors for dendritic sodium spikes, and we do it in a two-step fashion. We first run a simulation twice. We have the same synaptic inputs with frozen noise. We repeat exactly the same history of synaptic activations, and we run it first with the full model with all the channels present. That's the active model. And then we run the same simulation with, say, dendritic sodium channels knocked out. And we look at the difference in local voltage between the full simulation in blue and the passive dendrite simulation in gray. And if that deviates by a certain percentage, we say there's an active event here. That depends on dendritic sodium channels. And then we relate this to an operational definition where we simply read out the local sodium conductance density and define a threshold based on the thresholds that we have set here, that has a good prediction of whether the passive and active simulations would have been so different that we would call this an active event. And from then on, we can say we detect a dendritic sodium spike just based on threshold crossings of the local sodium channel conductance. We can do the same with some more extra conditions for NMDA receptors. Here we demand that there's a certain current going through NMDA receptors. Again, we have an active simulation with the full model, and we have a passive simulation where we made the NMDA receptors ohmic. So we didn't allow magnesium unblock, we made them linear. And when we do so, we of course get rid of NMDA spikes. And the difference shown in yellow here is an indication of NMDA spikes having happened. And we relate those to the amount of NMDA current that we need to sustain them and the duration for which that current was present. So having these detectors, we can now play games with the model. We can look what happened each time in space and time before such a detected event. For example, here, we are looking at all the synapses that were active near a site in a dendrite where at time 0, the sodium spike detector said, here is a sodium spike. And we see there's lots of local synapses right where the event was initiated, but there's also a little tail of extra synapses that were cooperative and also are more frequently active than baseline. This is all baseline subtracted. to trigger the dendritic sodium spike. And one of the reasons we did this plot is to figure out what is the spatial-temporal context that's relevant, which are the synapses that could causally be thought to be the trigger synapses for the dendritic spike. And so we will, in the next slide, count all the synapses in this red rectangle here. Likewise, for NMDA spikes, we have a detector for NMDA spikes, and we look back in time and we look across space to see which synapses were active when this NMDA spike was triggered. And here the answer is that these synapses are mostly local. This is in part because we demand that there must be an NMDA current through the local synapse. So we may have restricted the region of synapses that contribute to our detection of NMDA spikes. Again, we count all the synaptic activations in the red rectangle. And when we do so, we find that there's surprisingly few synapses active in these rectangles. So in the rectangle that is triggered by the NMDA, by the sodium spike, we find on average 2.3 synapses. And if we move this rectangle to random times not associated with dendritic spikes, we have about 1.2. So the difference is on the order of one more synapse. And a similar picture emerges for NMDA spikes. So what this means is that it's only a few synapses that are really locally able to trigger a dendritic spike. Of course, they don't act in isolation. This is not a brain slice. There is hundreds of synapses active all over the neuron in other dendrites that depolarize it and bring it close enough to threshold for evoking such dendritic spikes. So the distribution of how many synapses were active in the red windows ranges from 1, of course, but there's also up to 4, 5, 6 for sodium spikes, because they are somewhat cooperative. And for NMDA spikes, it's really dominated by just the one synapse that has to be there as part of the definition. So we have a small number of synapses that are able to evoke dendritic nonlinear events, let's call it like that. How does it look like in time and space? So here's a spatiotemporal view where we have plotted the highest membrane potential seen in a dendritic tree at a certain distance from the soma. So this is not showing all the dendrites, this is collapsing all the dendrites, and only the one that's most depolarized is determining the color here of the color plot. And so red means it's going to -10 or maybe -15 millivolts. What we see here is that the detectors for the NMDA spike in orange and the sodium spike in red have flagged the spike happened here. And these little white symbols indicate successive detection in the same branch at later times. So the initiation really happened where the triangles are. And we can then see that there was, at the end of a branch, a voltage transient that is not present when a single synapse has been removed in the simulation, namely this one. So it was really this synapse that broke the camel's back and triggered a dendritic spike. If we take it out, there will be no dendritic spike. So it's really, with the respect of this one synapse, it's an all or none event. Of course, it couldn't happen without the background, which is fully shown, of the synapses that are also activated elsewhere in other branches. And the synapses are indicated by color and size in terms of their weight. So the strong synapses here, the standard synapses are the small ones. If we zoom out and look at the bigger picture, we see that multiple such events are triggered in the run-up to an action potential that is triggered at the axon and then backpropagates to the soma and dendrites shown here. And some of these happen in the middle of a branch, and then they propagate towards the end of the branch and towards the branch point where impedance mismatch means it fails. Some of them are both NMDA in orange and sodium in red. Many of them are just sodium events, and they seem to increase in number just before the somatic action potential was triggered, which means that they might have contributed to getting the soma to produce an action potential. Here's a movie that will take some time to start, where we on the right show the same spatiotemporal plot, and we try to illustrate it on the left using actually an animation. So you can see all the synapses that are being activated as they are being activated in time. Again, they are coded by color and size for weight. So the largest ones are bright and large. They have up to 3 nanosiemens. And we also have the memory potential on the left. The dendrites have a color scale, and you can faintly, I hope, see local depolarizations that occur when there's a local event, for example, right here at the top left. So this will go through the, thanks. This will go through the movie. I will skip ahead to the next slide for lack of time. Oops, yeah. So triggering now on an action potential, we can look back in the same way as we did when we triggered on a dendritic spike event, and we can look back on how many dendritic spikes events we observed using our detectors in space and time. And you can see in the top that going back up to about 15 to 20 milliseconds, there is an increase in the number of sodium spike events detected that happen at 2 distinct distances from the soma, which is mostly due to morphology. There's simply more dendritic branches at these distance ranges. And so on the order of 6 or so extra spikes that we see before an action potential, and a similar number, but no, half that number roughly, for extra NMDA spikes that we see. What does that mean? One experimental observation that's puzzled us and others is that in vivo, when you block NMDA receptors in just one neuron, and you can do that by filling it with an intracellular blocker, MK801, you tend to block the action potentials from that neuron, even though the whole network around this neuron continues to function as normal and gets the same input. And so this finding, which was made by Laftzin et al. In 2012 from Jackie Schiller's lab, by Spencer, and by Lucy Palmer and in Matthew Larcom's lab in 2014, all these experimental papers agree that taking out NMDA receptors will stop the neuron firing. And we can quickly replicate that with our model. We didn't have to tune that, it just drops out of it. We can even replace the mean depolarization that is lost by injecting current at the soma, and we still get no output when you take out NMDA receptors. So NMDA receptors in their nonlinear form are responsible for, are necessary for all the output. You take them out, you make them omic, you undo magnesium unblock, and there will be no output. And another way of looking at the importance of nonlinear mechanisms for the output is to just ask, what if we take away synapses one by one and we start with the strongest in the model? If you do that in control, in black, you have to take out about 42 to reduce the action potential frequency by about 1/2. But we have to take out twice the number if NMDA receptors have been blocked and everything has been compensated by somatic current injection so that it does still fire at 0 synapses removed. So we can see that having dendritic active mechanisms enables fewer synapses to have more say on the output of the neuron, or any output at all, because if you don't compensate the block, the cell will not fire. And that also spills out to the tuning of the neuron, not just the fact that there's output, but also how well it is tuned, because it is the strong synapses that have similar tuning as the postsynaptic neuron. So to conclude, we've shown with simulations that NMDA spikes, but also sodium spikes, are an important component in generating any action potential output at all, in agreement for NMDA spikes with the experiment. We've shown that in a scenario where you have a realistic distribution of synaptic weights and selection of synaptic weights based on presynaptic neuron and postsynaptic neuron tuning similarity, it's a small number on the order of a few hundred maybe of synapses that can trigger dendritic spikes in vivo. And then once they happen on a sufficiently high baseline or in a sufficiently high number, that will trigger an output of the neuron. So what does this mean for artificial neural networks? It seems that the units that we should consider should have some nonlinear localized mechanism that corresponds to some form of dendritic excitability, such as that given by NMDA receptors or sodium or calcium or other voltage-gated channels, which mean that the summation of synaptic inputs is not completely linear. It's also not only sublinear as you would get with ohmic synaptic conductances. There is some nonlinear superlinear component that seems necessary for generating exponential output under the conditions of the standard in vivo brain, which suggests that these mechanisms are always engaged when our neurons in the brain are firing. So it's probably that these dendritic mechanisms are essential for doing what neurons do. Okay, thank you.
Thanks so much, Arnd. So that's a very nice detailed biological perspective of just how many spikes and active properties there are. We had one question already from the audience, from Taylor Carl at Emory, and then I think Yiota has a question. I think Taylor Carl has read a fair amount about calcium spikes in dendrites and cortex, and is very prominent in many models. You didn't mention them. So do they occur in your model? Are they different, not important? Can you not distinguish them from NMDA spikes? Or what's the relationship between your type of spiking and those calcium spikes?
Yeah, thank you. That's a very good question. So our model gave calcium spikes short shrift, unfortunately, but it does contain calcium channels. So if you added calcium dynamics to our model and you would simulate, calcium imaging readout, you would get calcium signals like people observe. It's just the calcium channels don't drive their own spikes in our model. That's, I think, a shortcoming. We really focused on simply sodium channels and NMDA receptors in particular. And in reality, all of those dendritic spikes always involve all those channels, just in different proportions. So an NMDA spike will also, of course, have a calcium component. That's the way you can observe it. And a calcium spike will be boosted by NMDA receptors present. Blocking them will make it harder to observe the calcium spike. But you're right that this model is, of course, not the full detail. It has abstracted away the calcium spikes.
Well, thank you. Yiota, do you have your question, or was it the same?
Actually, it was, I think Bartlett was first, and then Xiao-Jing, and maybe afterwards it's me. So
okay.
Thank you, Arnd. I really enjoyed your talk. So I had a couple of questions, but I'll maybe ask one and then let someone else cut in. So I noticed you didn't show a voltage movie of the dendritic tree. I was very curious about sort of the spatial distribution and like how finely articulated the voltage signal would be in the dendritic tree. You see at the summit, you see all these little spikelets of different heights riding on top of, slower things, presumably NMDA spikes. And I was wondering, because voltage is sort of smoothed out across a cable structure like a dendrite, do those very kind of complex-looking voltage traces nonetheless look? Can you see these hotspots? I noticed you showed where the synapses were in a very discrete way, but we didn't. I was wondering how discrete and articulated the voltage signal appears to be.
Yeah, I'm very happy about the question. I'm sorry that the movie is not better. So there is, in fact, voltage coding as color on the dendrite on the left. It's just that even though we cheated and made the dendrites wider, this is often very just a single pixel. And then with typical video coders, that gets smeared out. So if you do download the movie from the PNAS website, you may have a higher quality. And if you watch it, you will see exactly what you want, which is the memory potential spatial distribution on the morphology coded by color. And the answer to how crisp it is very localized if a dendritic spike or event happens close to the termination of a thin dendrite. Then, of course, the space constants are very short, and you can have a spike that is only a spatial extent of 20 microns. Very much like what the Houston paper showed in voltage imaging, for example, or what an NMDA spike looks like in Jackie Schiller's original 2000 paper. We also have wider events, especially if multiple synapses cooperate. So a sodium spike in a longer branch that is driven by 3 synapses can have 50 or 100 microns extent.
I think we have time for one more quick question from Xiao-Jing, and then unfortunately we have to move on to the next one.
From who?
Yeah, you're up next, I think. Yeah.
Sorry, Bartlett. Maybe let's discuss offline. Or wait. So Dieter, how should we do it? Should I answer more questions from Bartlett?
No, I think
Okay.
Xiao-Jing had a question, but he's muted,
muted.
so we can't hear the question.
I'm not muted.
I can.
Call on somebody, Dieter.
Thank you. My question is about different pathways. For example, there's some evidence that local synapses, recurrent connections within the area tend to be localized in basal dendrite. What do we know about heterogeneous distribution of NMDA receptors for different pathways? Roughly speaking, you have three kinds of connections: local recurrent connections, feed-forward long-distance connection, and feedback connections. Is there a way to see whether the NMDA receptor distribution somehow depends on which pathway actually we look at?
Yeah, I agree. So our model does not specify which pathway an input comes from. We conceptually think of most inputs to arise from other layer 2-3 neurons nearby. But of course, there will be inputs from bottom up, from layer 4, for example, and there will be top-down inputs that come from layer 1, from different brain, different cortex regions. Our model is not specific to that, but what I can say is that the way we try to tune our NMDA ampere ratio and the properties of the NMDA receptors that we do use, that is based on papers that studied connections from layer 4 spiny neurons projecting up in a columnar fashion to layer 2-3 pyramidal neurons. So Feldmeyer and colleagues, they patched pairs and characterized the NMDA receptor voltage dependence and kinetics. And that's where we took our NMDA receptor, part of it, description from. You're completely right that other NMDA receptors for other synaptic pathways might be different, but I think we are in part lacking the exact data to how they would be different.
All right, unfortunately, we have to move on, but that is a great start of a discussion. So we can hold some of the remaining questions for the general discussion. I think we're already set for a really interesting general discussion as we keep moving. So Brent, please share your screen, and you're on.
All right, can you see my screen?
Yes.
Great, perfect. All right. Thanks for inviting me. Every workshop needs a villain. And I'm not going to talk about dendrites today. However, I chose the talk that I'm going to give because it's going to be motivated by a data set. You're going to see where we're talking about cell-specific inhibitory plasticity. This is a novel data set collected in collaboration with Emory Oswald. And I've chosen to model the consequences of this data set in a point neuron sort of network framework. But there are going to be clear other paths I could have taken where dendritic aspects could have been chosen. I think this could be food for discussion during the panel here. I'm going to be talking today about assembly formation in networks of spiking neurons. So here I'm going to have some population of neurons, and I'm going to want to think of things in sort of a mean field framework. So I'm going to think about sort of the average activity and the average connection strength. And I want to think about a pair of excitatory groups. And I want to build in an assembly. And what an assembly for me will be is a scenario where the synaptic, the scaling weight W here is going to be greater than 1. If W is greater than 1, you're going to have stronger within assembly connection than between assembly connection here. It's sort of well known that if you start cranking W up here, even if you have inhibition, and if inhibition is non-plastic, so if inhibition is just a static connection here, you can increase W, strengthening the assembly further and further and further, but up to a point. And then you get to a scenario where the system's unstable and you get extremely sort of pathologic rhythmic behavior in the system. It's been sort of well known, Tim Vogel's, along with Wolfgang Gerstner, sort of showed that if you have the right kind of homeostatic plasticity mechanisms in inhibition, then you can fix the firing rate of the excitatory neurons, even though the connection strength is increasing significantly, and you can embed an assembly in the system without the fear of sort of runaway activity. So here I'm going to want to sort of extend this. So let's play the game where now I have my two assemblies I'm going to make inhibition assembly specific here, and I'm going to have that the inhibition within the assembly block here is stronger. So the I1, the inhibitory group I1 projects strongly to E1, same for I2 to E2. If we start increasing the assembly structure here, we see that we retain sort of asynchronous dynamics here. So even though we have homeostasis, we pay a price in the sense that we don't see the assembly structure that we're embedding into the system by increasing W in the dynamics of the response here. So the other way to do this would be to have lateral inhibition. So here now I1 inhibits E2 and I2 inhibits E1 strongly. And if you do this, you can definitely see the assembly structure, but it comes at the cost of it being sort of, I would argue, almost pathologic in models. It's very easy to get this extreme winner-take-all behavior that doesn't really look a lot like what people record in the cortex here. So we're sort of sitting at this dilemma. If we have sort of homeostatic inhibition, we can have a nice asynchronous stable system, but we don't see the assembly structure and the dynamics. On the other hand, we can see definitely see the assembly structure here with lateral inhibition, but it's pathologic here. So the take-home message for the talk here is, along with my collaborators, we thought that the rich diversity of inhibition, namely the different cell classes, is going to also support diverse inhibitory plasticity. And this is going to allow some coexistence between homeostasis and competition here to sort of navigate around that dilemma I just put forth here. So first I'll show you the data here. So this was data collected by Marta Kentos-Bustos in the lab of Anne-Marie Oswald when she was at the University of Pittsburgh. Now she's at the University of Chicago. And what Marta did is she looked at slices of orbitofrontal cortex. And what she could do is optogenetically activate a specific inhibitory cell class, record with a patch electrode in a pyramidal cell here, and then we can record the IPSP strength for various, and you can force the E cell to have a spike as well, and do this for various lag and leads for the plasticity protocol. This is going to be a spike timing dependent plasticity style protocol here. If she activates PV neurons here, and if we look at lags or leads that are either within minus 30 milliseconds to 0 milliseconds or 0 to 30 milliseconds, it doesn't matter. Both of these protocols lead to an overall potentiation of inhibition. So this is what I would sort of call a homeostatic style inhibition. It doesn't really care about the timing relationship, pre versus post. It really just cares about the coincidence here. If we push it further, then the plasticity kind of goes away here. So if they're firing far apart from one another, plasticity decreases. So this is the sort of classic homeostatic inhibition that several people have found already in the cortex here. If we switch it up and now do the same experiment, but with SOM cells, we get a different story here. So we see this sort of Hebbian style asymmetry here. And if we're in this region here, this is the region where the SOM cell fires before the pyramidal cell here. So the pyramidal cell lags the SOM cell. If that's the case, the SOM cell increases its inhibition and potentiates. If on the other hand, if the pyramidal cell fires before the SOM cell, we end up getting the depression of the inhibition, the recurrent inhibition back onto the system here. And of course, if you go farther out, you basically get nothing. So there's this differential plasticity of inhibition when we're considering PV neurons and SOM neurons here. And I'm going to want to explore the consequences of these different plasticity rules in a spiking network model where the goal is to embed assemblies into the system. So this was done by my postdoc, my ex-postdoc here, Freshta Lagzi here, where she took a spiking network model and sort of, I'm going to build it up here as we go along. What she did is she constructed a network where all of the excitatory connections were plastic with a standard sort of spike timing dependent plasticity Hebbian rule. So all the E cells, whether they're part of assembly 1 or assembly 2, right now there's no assembly. I have to train the assemblies in. This is just a group of excitatory cells. All excitatory synapses have plasticity. We're going to put in a group of PV cells. They're going to have the homeostatic style plasticity that Marta found in orbitofrontal cortex. And we're also going to have SOM cells here that are going to have the Hebbian style plasticity that Marta found. And I'm going to separate two groups, an S1 and an S2. And the reason for that is I haven't laid down the non-plastic connections yet. So all the connections you see here are plastic with their respective colors. So now I have to put the rest of the connections in here. And all the black arrows are non-plastic. And what's the reason why we have S1 and S2 is we're going to walk in with a tacit assumption that this group of E1 neurons, even though they're not in an assembly yet, they're going to project with higher in degree or effectively higher probability to S1. So in some sense, tethering S1 to E1, same thing E2 to S2 here. So we're going to hardwire that into the initial adjacency matrix. These are non-plastic synapses. And then we're going to subject these kinds of networks to a training protocol here that I'll explain in a second. Okay. So first, I'll have a control network where I've removed the SOM cells and I've replaced them with PV cells. So this is sort of the classic homeostatic plasticity framework here. The way we're going to train the system is all the neurons in the E1 population are going to get some external correlated fluctuations that tend to synchronize these. So you can see here before training, their spike train activity is relatively asynchronous. During training, we're starting to enforce correlations through an entrainment in the system. And then we're going to ask what happens after training here. And in this particular framework, we don't get much. We try to train the network, but then when we stop training, we go back to this asynchronous solution, sort of like I was talking about at the beginning. If we look at the correlations within pairs of cells within the assembly, they're pretty weak. If we look at correlations between cells, so a cell in one assembly and a cell in a different assembly, they're also pretty weak here, okay? So let's now compare that to the network I want to study here, the one with the plasticity rules that Marta found. During training, we again get strong correlations, but then after training, this correlative structure persists and persists indefinitely in these models. And we see very strong within population correlations and very strong anti-correlations between populations here. So this network produces a rich dynamic here of competition that doesn't look pathologic. It's not this strong winner-take-all framework, but it persists for a very long time after training. So let's unpack this and ask, why did this happen? Okay. Well, to do that, I'd like to sort of dig up older theory of how to think about spike time independent plasticity from Kempner, Gerstner, and Van Hemmen. So what they said is, if we know the correlation function between a pair of neurons, this is what CS is. And here I'm just going to add the shelf of firing rates that they typically sit on. So if C of S is 0, then the neurons are completely asynchronous. C of S here being non-zero is some temporal structure in their correlations. The evolution of the weight is effectively, oh, I'm missing a term here. Oops, I'm sorry, I did this quickly. There should, in this equation, you should also have the plasticity rule. So this gray curve here is my plasticity rule. So in order to look at the evolution of the weights, you have to multiply the plasticity rule times the correlation function, and you have to integrate over time. And then that's looking at all possible pairings of pre and post, weighting them with the appropriate synaptic increase. And then that gives us the overall evolution of the weights. And really what matters is the area under this. And in this particular example here, so you have to sum U1 and U2, sort of this area and this area. If this is greater than 0, then the synapse will potentiate, because this thing will be positive, so the synapse will grow in strength. If it's negative, then it'll depress here. So this is an old theory that has worked well at sort of understanding these simulations here. So what's happening for these two connections during the training phase? So here I'm going to look at the within assembly connection in blue, the cross assembly connection in red. And if we look at what's happening here, since there's strong correlations within the assembly during training and even after training, if you multiply this correlation function times the SVP curve and then look over time, you just get overall potentiation. So you get a strengthening of the within assembly connection here. Since there's anti-correlations between the neurons in adjacent assemblies here, you get the opposite effect here. So the integral of the plasticity rule times the correlation function tends to be negative here, and then between connections tend to decay here. And this is what's giving us our assembly structure in our network. That's what's happening for these connections. So how is that actually happening? Why are we getting strong correlations within assembly that drive potentiation and strong anti-correlations between assemblies that drive depression here? So now let's look at these synapses here. So what ends up happening with this plasticity rule is the mutual connections. This is the SOM cells projecting back to the group of pyramidal cells that tend to drive them. They tend to lag because they're being driven by these pyramidal cells. Their spikes tend to lag the spikes of these pyramidal cells. And since that's the case here, when we overlap it with this plasticity rule, overall we get depression. So this connection going from SOM1 back to E1 tends to depress. And here, I'll remind you, this is negative here. So depression means going upward in this scenario. On the other hand, if we look at the cross connection here, this ends up having an integral that's positive. So overall, you get potentiation. So what this plasticity rule for SOM cells is doing is it's carving over time this competitive lateral inhibition in the circuit. I won't show you here because I don't have time. The PV cells do sort of the classic homeostatic rule here, and they tend to potentiate onto groups of neurons that have high firing rates to control their firing rate. But it's really this SOM cell inhibition, or the plasticity of SOM cells, that produces this lateral inhibition that gives us the anti-correlation between assemblies that depresses connection and then produces correlation within assembly that potentiates connections here. So let's to finish off here, let's build a mean field theory to sort of understand this. So I've done this with several postdocs and grad students in my lab, but sort of this particular version is in our paper with Foresta and Marta here. So, and I'm going to abuse the fact that if I freeze the weights, if I freeze the weights in the network, then I can use a linearized theory to, if I know the structure of the network, I can compute what the correlations in the spike trains are, because this happens on a fast time scale. On the other hand, I just showed you the Kempner formulas that if I know the correlation function between neurons, then I know the evolution of the weights, and this is going to happen on a slow time scale. So I'm going to use this classic applied mathematics trick of having a fast, slow time scale separation to build sort of a mean field theory for this. And here's sort of the equations. Again, for frozen weights, I use a linear theory to talk about how if I know what W is, I know what the correlations functions are. And on a slow time scale, I know what the evolution of the weights are because I know what the correlation functions are. And I solve these in a self-consistent way. And I don't have time for this, but you can write down the evolution equations for the weight of the connections within the assembly and the weight of connections between assembly. And here I'm assuming symmetry. So I can ignore the other two connections here that I'm ignoring. I'm really interested in the connections within the excitatory group, but this mean field theory also includes the inhibitory cells here. We can use sort of tricks to reduce the dimension of this to effectively a two-dimensional system, which will be nice for phase plane analysis. So here's what the phase planes are for sort of two of these networks here. I've gotten rid of the two inhibitory subclasses and just had one for ease here. This is the vector field during training. This is during training here. And the blue curve is the null line for the growth, for the weights of within here. So if I'm to the right of this blue curve, then WW here increases. And if I'm to the left of it, it decreases here. And in this network, there's a large real estate in this particular parameter regime where we get overall depression. In this network, on the other hand, there's this sort of larger real estate here where we get overall potentiation of the within assembly connection and a depression of the between assembly connection. That's exactly what we want, but this is during training. The training is sort of enforcing this to be the case because I'm giving lots of correlations to neurons that are within the same assembly, and I'm not giving any correlations to neurons that are between assemblies. So I'm sort of enforcing this structure here to start with. What I'm really interested in is what happens when I'm done training. What does this phase portrait look like here? And this slide's going to be complicated. It's a little ugly, but I'm going to simplify it in a second here. So here's when I stop training. Now it's only internally generated dynamics in the system. What I'm really interested in is I want to find the zone where I get an increase in this synaptic weight and a decrease in the between synaptic weight. Now I'll help you here. I'll color those zones in gray here. So if I can get this, so if I'm in the gray region here, I can have a stable assembly in the sense that I've stopped training, and yet I'll have an increase in this synaptic weight in terms of within population synaptic weight, and then between population synaptic weight will decrease. And actually, I'm sorry, these should be inverted here. Sorry about that. This gray zone is much, much larger for this for the assembly for the network with the two different cell classes here. So it's much, much easier to train the system to get in this gray zone. And once you're in the gray zone, then the assembly will stay around even after training here. So this is what this rich diversity of inhibition is doing, is it's increasing the sort of the zone where we can have stable assembly learning here in this network. So to summarize, what I've shown you here is I've shown you data that shows there's differential STDP from PV cells and SOM cells onto principal neurons. This is an orbitofrontal cortex. The PV plasticity supports homeostasis. The SOM cell plasticity supports assembly competition. And a mean field theory shows that when you combine these two things together, it's much easier to get stable assembly formation with less training. What about dendrites? So in the last sort of minute and a half, I'll explain sort of my thoughts on this. I started off with this data set here that has this, this differential plasticity. And then I started modeling. And I chose to go the direction of saying, well, I want to talk about networks. So I'm only going to care about connections. And it's, for my purposes, fine to use point neuron models, really overly simplified point neuron models. But maybe if I was a hardcore dendrite person, I would have said, well, wait a minute. We know that PV cells tend to project on the somata of pyramidal cells, where SOM cells project on the dendrites here. And I guess the question I would have is am I missing an opportunity? Is there a different narrative or a different set of predictions I could have made had I taken this this well-known observation of inhibitory circuitry into account? Or is this going to matter at all? So I chose to go the network oversimplified reductionist network direction, but there easily could have been a separate story along this path here. So with that, I'll close. I'll thank my collaborators. So the modeling was done by Freshta Lagdi when she was in my lab, and all of the experiments were done by Marta Kentos Bustos in the lab of Anne-Marie Oswald. And I'll thank you guys. Great.
Well, thanks for the network perspective on plasticity and interneurons. Any questions from the panel for Brent at this point? Yes, Yiota.
I mean, thank you, Brent. You already mentioned it towards the end that the projection sites are very different for the two types of interneurons. So what do you think would change in your story? What would be your expectation if you accounted for apical dendrites and especially their nonlinearities?
Absolutely. So I think there's several data sets actually with the same collaborator, Anne-Marie. She's also got some interesting data where SOM cells can control whether pyramidal cells will potentiate synapses amongst each other, because those synapses are located on the dendritic tree, and SOM cells can inhibit the dendritic tree. So by effectively if you inhibit the SOM cells, removing their activity, that's the recipe for getting strong pyramidal cell learning here. That doesn't require the synapse to be plastic between SOM cells and pyramidal cells, but that site of SOM cell inhibition is a gate for excitatory learning. And that's at the core of assembly formation. I really care about the E to E connections and whether they can be learned or not. And Anne-Marie in the same set of experiments, well, not the same experiments, but the same brain region with the same postdoc actually, they've now shown that SOM cells can gate pyramidal cell learning. PV cells not so much. It's harder for them to do that, and it's probably because of the location on the tree. So putting this all together is something we've been talking about, but one step at a time. But that would be my thought, is that I'm missing an opportunity that SOM cells, because of their location, can really control pyramidal cell learning, and that's not part of my story, and maybe it should be.
Thank you.
We had one question from the audience from Ilya Nemelman, a bit outside of the scope of the talk, but do you have an intuition respective, what would happen in a random balance network with and without diverse inhibition on the same train washout as you explored?
So say the last part again. So,
yes, in a random balance network with and without diverse inhibition under the same train washout as you explored.
The same what washout? I'm not quite.
Yeah, this is the downfall of just having the little questions to read off.
So if you try to train in assembly structure, so balance networks in what Ken Miller calls the tight balance regime. If you try to embed structure in the excitatory population, try to embed assemblies in the system, and you don't allow inhibition to be matched to those assemblies, then the network will quickly go haywire. And that's because what's in a balanced network, inhibition's job is to track and cancel runaway activity. If you've got two assemblies that in principle are running away through different wiring, then they need compensatory inhibition in order to make sure the system remains stable. That's why I went from just a single inhibitory population to a pair of inhibitory populations, right? In order to make sure that the system was always stable. If I just had a random balanced network that has just one inhibitory population, and I don't have interesting plasticity of inhibition to help that, then I'll quickly just blow up. I can only embed very weak assemblies in the system before it starts to go haywire.
Okay, thanks. So this is a great point to keep in mind for the general discussion. We have network computational questions on inhibition, excitation, interactions, and then what do dendrites do when we go to that level of complexity of network interactions? And I think we have a lot of cool stuff to discuss in the general discussion. At this point, to stay on time, we should move on to Dr. Bartlett Mel from California.
Thank you, Dieter. Let me get my, share. Okay. Oh, hold on, sorry. Okay. Okay. Yeah, thank you very much. And happy to be here, Dieter. Okay, so actually, let me go back to my title, How to Optimize a Dendrite for Binary Subunit Pooling. Okay, so what is binary subunit pooling? To talk about that, I'm going to spend a couple minutes talking about a problem that we looked at previously in earlier work that kind of motivated this. This is a picture of a basic neural memory structure, kind of an old-fashioned one. You have inputs coming in, these axons coming in from the side, making contact onto the dendrites of a population of neurons and making synapses here and there on those neurons. And this could be like CA3 projecting into CA1. This could be the lateral olfactory tract projecting into the piriform cortex, where changes in these synapses are going to be encoding kind of one-shot new memory formation. So we've previously studied this type of network, and we've asked the question, well, okay, if a cell is not a single point neuron, if each one of these neurons is not a point neuron, but has separately thresholded dendrites that can act as signaling and learning units, then how much does a network with cells like that benefit from that, from having dendritic subunits compared to not having them? This kind of study that we've done. So let me just sort of bore down a little bit into this model that we looked at previously. So here's those inputs coming in from the left. Here's a bunch of neurons with their dendrites sticking up and receiving contacts, synaptic contacts, strong and weak contacts. And basically what we viewed this network as being was a bunch of dendrites and the cell bodies. So there's a cell, and the cell has several dendrites or a few dozen dendrites, but the cell body was sort of irrelevant in this model that we looked at. Basically what happened was that the dendrites would through synaptic changes would fire, and then the cell bodies would collect those responses. And if this neuron here had one of its dendrites firing, then it would fire bursts of action potentials and pass it on to the network as a whole. And the response of the network as a whole was essentially a sum of all the dendritic activations. Okay, that's how we characterized the response of the network. And basically what you would want to have in a memory like this is when an input pattern comes in and the synapses change, then as a result of those changes, when that input comes in again, there's greater activity in the network. There would be more dendrites firing, and then there would be more neurons firing, and then there would be a greater network output, and thus the pattern would be recognized because it had been previously trained. Okay, so I want to focus in on the job of one neuron because we're trying to get to this notion of binary subunit pooling. So here's just one neuron, and it has, again, several dendrites, and these are the synapses that are formed from these axons coming in. And so an input pattern could come in, and these 4 red axons are the ones that are firing, and they're activating synapses on different dendrites, some of them strong, some of them weak. Another pattern comes in, nothing happens. Another pattern comes in, and still nothing happens. And eventually something's going to happen in this picture, which is that a particular dendrite gets a particularly strong, by accident, by random connectivity, gets a particularly strong activation pattern shown here, for example, in here. And there's a learning threshold on this dendrite, and the dendrite goes into a learning mode. So a threshold is crossed, not necessarily a firing threshold of the dendrite, but a learning plasticity threshold. And as a result of that, then any of these synapses that are active would be potentiated or refreshed. Anyway, some of them were strong, but as a result of this, all of those synapses would be potentiated. And now they're all sort of shown as big circles. And so if patterns come in again, and later that particular pattern comes back, now since all of the synapses are strong, then that dendrite is now going to actually signal and fire, so to speak, fire a local dendritic spike, and that will be passed on to the cell body, and then that will be passed on to the network. And this would be the way that the network would know that this neuron had recognized, or this dendrite in this neuron had recognized a little piece of the input pattern, and that's kind of a little piece of the recognition thing that has to happen. Let me go to the next picture. So the point about this is that this neuron's job, what is the job of the neuron? The job of the neuron is to respond definitively if any one of its dendrites, and it might not have, I'm showing 5 here, but a real neuron might have, if this was in CA1, maybe it has 20 or 30 dendrites in stratum radiatum, or if it was a human neuron, maybe it has 50 or 80 dendrites in its basal dendritic tree. But if any one of them goes off, then the neuron has to reliably generate a burst of action potentials to signal to the downstream neurons. And if none of them fires, if they all are receiving threshold input, then the neuron should definitively not fire. And so there's this game where the neuron has to generate a very different response if even one out of many dendrites is firing, even though all of them are receiving input. And so basically we want trained response to trained patterns to be reliably larger than the response to untrained patterns. So this, and so that there can be a threshold placed in between them, and then the neuron can reliably signal that one of its units went off and say yes, if not, otherwise say no. Okay, and those are false positives. Now, so we're asking a dendrite to act like a binary signaling thing. And so this is challenging. This seems so easy. If it was an electronic circuit, then this would be like an OR gate with many inputs. If any one input goes off, then the neuron would signal. But the problem is a neuron is dealing with a lot of continuous variables. And this is kind of what we ran into. And this was like making smoke come out of our ears because we were trying to figure out how this would actually work. So think about it this way. Suppose we had even the simplest thing you could think of would be that you're going to stimulate a dendrite over and over again with the identical kind of input, namely every synapse has an equal probability of firing. It either does or doesn't, and you're going to get patterns of activation. And so, for example, imagine this dendrite that has a bunch of spines on it, and on average, 10 of them are active. It's like a binomial process where every synapse just flips a coin and it fires or it doesn't fire. And here we get an average number of spines activated at these locations. But of course, there can be a variation in how many if you play patterns over and over again, sometimes you're going to get more synapses activated, sometimes you're going to get less synapses activated. And this is sort of continuous variation in the amount of activity, irrespective of location. But then there are location differences. Sometimes the inputs will fluctuate distally. Sometimes the inputs will fluctuate proximally. So these are all 8 synapses. Sometimes, and distally activated synapses might be more effective at triggering. They are more effective at triggering dendritic spikes, but proximally activated synapses are more effective at delivering current to the cell body. So there's a lot of variation that could arise from nonlinear interactions or nonlinear effects of spike triggering and attenuation and things like that. And then there's another kind of variation too, which is that the synapses that happen to get activated on the dendrite could be sort of more diffuse, or they could be more clustered. And when synapses are more clustered because of NMDA channels, they can deliver more NMDA current. And so you have a lot of continuous variation. This does not look like a unit that when you hit it with these nominally the same kind of input over and over and over again, that it's going to generate either 0 or 1 outputs. Okay. And this would be true whether we're talking about spike, whether something triggers a spike, one of these patterns does or doesn't trigger a spike, or whether it's a subthreshold. There's still going to be a lot of continuous variation. And this is working against this idea of signaling kind of binary fashion. Okay, so let me go back here into this picture and say, what could the brain do to mitigate this problem of lots of variation in the response to sort of similar statistically similar input patterns? So a lot of the variation that we're looking at in this picture has to do with the locations of where the spines are that are activated. And so one of the things that would influence the statistics of where spines are activated is where are the spines? And so this says that we should be looking at the spine density profile across the length of dendrites. And so if you do that, there are a couple of papers, there's not a lot on this, but there's a couple of papers. One of them came from Nelson Spruceton's group. And they looked at the density of spines near the branch origin. This was in CA1. And they looked at the density further near the branch end, and there was a significant drop off in the density of spines from one end of the dendrite to the other in the hippocampus. And then also in the cortex, this is from Dielston. These were different neurons in the visual cortex in different areas, V1 and prefrontal cortex, and actually prefrontal cortex, and then this is in the infratemporal cortex. And there was this pattern. There was sort of a blank area near the base. This is spine density here, a blank area near the base of the dendrite. But then over most of the length of the dendrite, the spine density would fall off continuously, regardless of the visual area. So this is interesting because it maybe would impact on the variability, spatial variability, or spatially induced variability of dendritic responses, which is related to the problem that we're asking. Okay, so we worked on this problem with Tejas Ramdas during the pandemic. He was an undergraduate at USC. He's now a graduate student at Harvard. But anyway, this is his project for a year and a half. Okay, so we were going to focus on the effect of the spine density profile on this binary subunit pooling problem. Okay, so the first thing we did was, here's a compartmental model. This is a ball and stick model, soma and a dendrite that's 200 microns long. And we're going to activate the spines along the length of it. In this particular case, the density is uniform of the spines. And this is showing a bunch of individual runs in which there were, in each case, just sort of to make a controlled picture, 22 spines that were activated, and these green dots were showing the locations of them. And you can't really tell by looking at this what any given pattern is going to do, but this is what they did. So the one on top here is going to be this trace on top, and the one on the bottom is going to be these traces on the bottom. So sometimes local NMDA spikes were generated in this dendrite, several cases of that, and then the rest of the time there were subthreshold responses. So you can see a difference between firing and not firing here. It's not exactly binary, but there it is, and that's for a uniform distribution. Now, so what are you going to do? We're going to try three things: uniform, increasing, decreasing. So as a kind of a control, we looked at the increasing distribution, meaning that the density is increasing towards the tip of the dendrite. And here you can see that there's sort of a rightward bias in where the spines are going to be activated naturally, because that's where they are. And it's sort of the exact same deal. Now, the responses that are generated, these are all, by the way, at the cell body, are kind of a similar pattern here, but they're smaller, basically. They're lower in amplitude. So these were going up like 20 millivolts from rest, and these are going up more like 10 millivolts from rest. Why? Because most of the synaptic activity is sort of biased towards the end of the dendrite, and so there's a greater amount of attenuation of the signal coming back towards the cell body. But nonetheless, a similar pattern. Now, with the decreasing spline distribution, this is sort of the one that is more realistic. You can see it's kind of a mirror image of the increasing case. The spines that get activated because of the higher density near the branch origin tend to be near the branch origin, and you can see those patterns here. And again, we have a spread in the responses, the superthreshold responses, and a spread in the subthreshold responses, but we have a much larger amplitude pattern here in this case. Okay, so what is the implication of this? Let's carry it through to see how dendrites that had those three different spine density distributions would perform at this binary subunit pooling task. So here's where you can't just look only at one dendrite. You have to look at all the dendrites together in the neuron. Okay, so here's our kind of imaginary neuron that has 30 dendrites is the number that we assumed. All of them behaving just like the dendrites that we were looking at and simulating. And we're going to assume that that one dendrite will receive one of these superthreshold inputs about 0.1% of the time. So 1 in 1,000 is the probability. And so pretty much on one neuron, you're going to only ever get one that's firing or not. Okay. So what is a case that the neuron has to distinguish? Well, the no cases when the neuron is not supposed to respond are ones in which all 30 dendrites are firing in the subthreshold range, which means we want to go to the subthreshold range of responses of those dendrites in that neuron and draw 30 of them, because all 30 are subthreshold, and add that up at the cell body and say that's the total amount of current, let's say, arriving at the soma. Now, in a case that's a yes case, we would have 29 of those subthreshold responses. So we would draw 29 from this distribution and 1 from this distribution, add those up and see how big that total is, and see if we can distinguish yes cases from no cases. So here is that. So the superthreshold cases are ones where we drew, again, 29 from the distribution of 29 from this distribution of voltage responses to subthreshold. So these are like 5-10 millivolts off the baseline, and that's this distribution here. We're going to draw 29 of these, and I'm sorry, this is the yes cases, the no cases. 29 of these and 1 of these from the superthreshold case, and add them, and versus the no case where we're going to draw 30 of these and add them. Okay, and then we're going to. This is the result of that. So the no cases are when you draw 30 subthreshold draws from all these dendrites, which are supposed to be putting out nothing. You wish. When they're not firing, but they're not putting out nothing, they're putting out this amount, and 29 or 30 of those added together creates this gray distribution. When you only have one dendrite going off, which is drawn from this distribution, you add 1 of those to the 29, and then you get this green distribution, which you can barely see here because they're rare, but it's also very mixed in with the gray distribution. They're not well separated at all. And if you wanted to draw a red line as a threshold between them, and the way that we just picked that red line is we said, let's make sure that 95% of the true yes cases are to the right of this red line, then we end up with this, we can make a precision recall curve. And this is where the precision recall is. Here we are at 95% recall, meaning we're going to catch 95% of the yes cases. But if we put the threshold there, then we're going to get 21% precision, meaning that we have almost 80% of the time when we say, yes, that was a yes case, we're wrong. Okay, so this is abysmal. The neuron would be essentially incapable of performing a binary subunit pooling task in this case, in this situation, because we can't tolerate 21% precision. We want like 90% precision or something like that. Okay, so that's that case. Now, what about the increasing case? I'm going to just throw these graphs up here for you. So it's the same. It's bad also. We have strong mixing of the yes and no cases because there's not very great separation between the superthreshold and the subthreshold responses. And we're, again, just by sort of luck at 95% recall, there's 21% precision if you had a neuron configured in this way with increasing spine density. Okay, so then the third case, not surprisingly, will be the decreasing case. Now here we're drawing 30 from this distribution gives the no's, or 29 from this distribution and 1 from this distribution gives the yeses. And now they're much better separated. And you can see that in this case, the precision and recall at 95% precision, we have 85%, I'm sorry, 95% recall, we have 85% precision, which is massively better than the than the other two cases. Okay, so so that's our first message, is that at first glance, a decreasing spine density distribution is way better for this task of sort of binary signaling. Yes, no, from an individual dendrite to the neuron, which has many dendrites. Okay, so we did a bit of analysis to try to understand why this is. And here we came up with a formula called goodness. In other words, goodness for this task of a dendrite. And it's pretty simple. It basically just says you're good if you have a big difference between the superthreshold magnitude and the subthreshold magnitude. So between spike, having a large spike compared to the subthreshold range, or you can look at that same thing here, the distance between this distribution of superthreshold responses, like up here around 30, 40 millivolts, compared to here 5, 10 millivolts. That distance is good to have, and that's, you can call that the spike amplitude. And then the other thing that matters is the distribution, interesting, of the subthreshold distribution. So you do not want a lot of variability in the subthreshold range when the dendrite is not firing. That's the distribution width, essentially, of this sigma sub. And that's because we're adding up a lot of those. There's a lot of subthreshold dendrites, most of them, 29 out of 30 or 30 out of 30. And the variability of this distribution adds when you add all of those random variables together. So we don't want a lot of variability there, because that's what washes out the difference between the yes and the no cases. And we made up this measurements because we were trying to figure out, well, how do we measure things about different dendrite cases and decide how good they're going to be, or try to predict it. And it turns out that this measure, if we just apply it to the cases that I already showed you, is about 50% bigger goodness for the decreasing case than either of the other two cases, so that it predicts that it will perform better at the binary subunit task, and it does. Okay, now why is it that the decreasing case is better? Okay, so this is a graph that basically during the course of a simulation, and a bunch of synapses are going to be activated in a burst on the dendrite in some pattern. And then this is a snapshot of the spatial distribution of voltage inside the dendrite across its length at the moment when the highest voltage peak is reached anywhere in the simulation, anywhere in the dendrite. So one of these, and then that spatial profile is just sort of put down here, and then hundreds of runs are done, and you can see hundreds of cases. And so basically what happens when you have a spine density sort of leaning towards the base of the dendrite is that the voltage response tends to be peaked near the base of the dendrite. And it's also quite consistent. That's important. So you have a biggest response, and it tends to be near the base of the dendrite. If you go to the uniform case, well, actually the increasing case is kind of the opposite. It's peaked. The voltage response at the peak of the voltage response in the entire simulation will be near the end. Not surprisingly, that's where the synaptic input is concentrated. But then of course, you don't see much voltage at the cell body or at the base of the dendrite. Now, the case of the uniform is not peaked very well at all. So because the point is that the synapses are activated all over the place, and you have peaks kind of all over the place too. So you have a lot less predictability about where the maximum voltage response will be inside the dendrite than you do in either of these biased cases. Okay, so let me pause here. I'm sorry, and I wanted to say, so this was the main effects that we saw was the increased predictability when you concentrate synapses towards a location, and in the limit, if you were to concentrate all of the spines in one location, then there would be no variability at all. And that would be perfect for doing binary subunit pooling. Unfortunately, you can't cram all of the spines into one location. You have to spread them out because, for other reasons. But anyway, cramming them together or biasing them creates less variability in terms of the spatial patterning of the responses. And then furthermore, in this particular case, the less variable part of, the source of the excitation to the dendrite is also closer to the recording, to the kind of the business end of the dendrite, where near the cell body or the base. And so it's getting a cleaner signal closer in. Whereas in this case, for example, you have a cleaner signal, but it's farther away, and the noisier signal coming from this low density of more scattered synapses is actually closer to the business end. So this is sort of doubly good to have the density concentrated near the base of the dendrite. So we looked at a few other things that are biophysical parameters and how they would influence it. I will just very briefly comment on this. The low spine neck resistance is good for binary subunit pooling. So this having high dots here at the 95% recall rate is good. And so the red curve is the decreasing, I'm sorry, this is the decreasing distribution, and this is increasing, and this is uniform. So the decreasing is better for low spine neck resistance and for high spine neck resistance, but low spine neck resistance is better for everybody. So that was another thing we noticed. And the thing that was really valuable for binary subunit pooling was having sort of a strong hyperpolarizing inhibition on the dendrite, something that would be sort of co-activated with the excitation, which creates a really nice separation between the suprathreshold and the subthreshold. This was actually a point made by Shoemaker, Pat Shoemaker, who wrote a paper 10 years ago making a point about this. And we took advantage of it and we remembered it and tried this. So that was like the miracle that made everybody perform well, regardless of the spine density distribution, was having strong hyperpolarizing inhibition. But we also realized that this was a ceiling effect because we weren't really simulating noise the way that we should have. And just very briefly, we said you can't count on having no noise at the cell body when you're trying to do this classification. So we added a kind of background noise at the cell body of a few plus or minus 3 millivolts. That's what we call background noise. And we also pointed out that different dendrites have different scaling factors. Like this far away dendrite might only have a scaling factor of 0.5, and this one might have a factor of 0.1. So this is introducing, depending on which dendrite fires or doesn't fire, it introduces another kind of nuisance factor. And we had that called dendrite coupling variability. But when we applied both of these realistic forms of noise, it lowered the precision at 95% recall for all of the densities, but the decreasing spine density profile was still superior to the others. So that was the constant that we saw in all of the experiments that we did. So here was our conclusion. We have the expectation now based on our conclusions that if you're a neuron that's optimized for this task of binary signaling, when you have lots of dendrites sticking into the cell body, you should expect to find well-isolated dendritic subunits. You should expect to find a decreasing density of dendritic spines. That's kind of the main thing I've been talking about. You should expect to find low to medium resistant spine necks. You should expect to find synapses with strong NODA currents. Now, I dendn't talk about this, but you should expect to find fast spiking sodium channels. That makes it work better too. And a deeply polarized or leaky dendritic membrane or prominent source of hyperpolarizing inhibition. So these are all things that you do kind of find in a lot of neurons. But I want to end just going back to this picture, which I love, because this picture says to me, when you can focus on the fact that they all seem to that in these different neurons in different areas have decreasing spine density distributions, but they're not all the same. In V1, it's very mild fall off. At the ends, this is because there's no spines near the ends of these dendrites, so you can kind of maybe ignore those, but there's a very mild density gradient here in V1, and a very pronounced density gradient here in the prefrontal cortex. And so we thought, well, that makes sense to us because in the prefrontal cortex, you're more likely to have neurons that have to do kind of binary decision-making tasks, whereas in V1, you'd expect more continuous signal processing tasks, and maybe they are not optimized for binary subunit pooling. We have no reason to think that they should be. So the fact that this density profile seems to be manipulated by the brain, and they're different in different areas, and maybe we can sort of in our heads imagine that there's a reason why you'd see a difference between, say, V1 and prefrontal cortex. We kind of like that idea of it, but of course it remains to be proven. And I just finished, I love this picture. It came from Teacher et al. This is a human neuron. I mean, humans, we simulated a cell that has a problem of dealing, like picking 1 out of 30 dendrites. We just came up with a number 30, not really for any good reason. If you look at the basal dendritic tree of this human neuron in the infratemporal, let's see, where is this? This is from the temporal cortex. I'm not sure exactly where it is compared to a mouse, but this thing has, I think I counted here, I don't know if I counted correctly, but like 70 or 80 dendritic subunits, and presumably is doing some kind of pooling over those units. So you have a serious problem of binary subunit pooling if you have this many dendrites. And we think, we expect, and I don't know the data, and I haven't seen this, maybe they have it, that we should expect to find a very pronounced decreasing spine density profile on these neurons if that's what they are involved in. And so that's kind of a prediction that we would make. Okay, finally, I just want to acknowledge Tejas again, who has now started graduate school. This entire thing I talked about is in a paper that's just about to come out, or maybe it's even available online in this special issue of Neuroscience devoted to dendrite papers. Oh, it doesn't have much information here in the title. So you're welcome to, I'll be happy to send that.
All right, well, good morning or good afternoon, depending on where you are, everyone. And thank you again for inviting me to this workshop. And in this talk, I'm presenting recent work that we have been doing in Chris Leismith's lab. And in particular, we are taking a somewhat simpler approach compared to what we've seen so far in this workshop, but it should also nicely connect to what we've just seen from Yiota. Our goal in particular is to analyze in how far passive dendrites can be systematically harnessed as a computational resource within large-scale neural networks that are built using the neural engineering framework. So the emphasis here being on passive in contrast to the active dendrite models that we've seen so far. So before I begin with that, I'd just like to give you a very quick overview of what the neural engineering framework, or NEF as I'll abbreviate it, is. And on a very high level, the NEF is a method for constructing models of neurobiological systems. That is, the NEF allows us to map descriptions of some desired high-level behavior that may, for example, come in the form of some mathematical dynamical system onto neurobiologically constrained spiking neural networks. Or in other words, this is a method that allows researchers to map functional or behavioral descriptions of biological systems onto neurobiological mechanisms. And to allow researchers to do this conveniently, there's also a software tool that's called Nango that implements the NEF as a relatively easy to use Python library. And this allows researchers to semi-automatically compile high-level behavioral models onto low-level spiking neural networks that are constrained according to neurophysiological data. And Nango has, for example, been used to construct, at least at that time, the world's largest functional brain model. And then there's like a sketch from that paper. At its core, the NEF is based on 3 principles, 2 of which we are using here in this talk. First, we assume that neuron populations represent vectors X within some relatively low-dimensional activity space, at least low-dimensional compared to the high-dimensional, to the high dimensionality of the activity space. And second, we think of connections between neurons as realizing arbitrary functions, well, of this form. Well, how does this work? So to represent variables X in a neuron population, we essentially assign a tuning curve AI of X to each neuron. That is, this AI maps an output rate to each represented value X. And we do this by mapping this X through a current translation function, Ji, and this outputs a current that then gets fed into a neural nonlinearity model. And this current translation function usually is essentially just the inner product between an encoding vector and the represented value plus some bias term. And the important thing here is that this is a normative tuning constraint. That means we never actually use this equation directly in our network, but instead we solve for synaptic weights that implicitly realize this tuning. And when we actually execute our network, we just have a spiking neural network with spiking neuron models that are connected through simple synapse models with temporal dynamics. And the crucial point here from a theoretical perspective is that if we choose these parameters carefully, then the corresponding tuning curves, which are depicted up here, form a universal function basis over a compact subspace of RD. And that means that as we increase the number of neurons to infinity, we can decode any function over the represented values. Now, given that the NEF allows us to construct universal function approximators, the question is, of course, A, whether it's possible to harness dendritic nonlinearities and somehow do better, and B, whether doing this actually helps us in any way, because as I said here, we are already able to construct a universal function basis. So from a population of neurons, we could decode anything. Right, and the important thing here, as I already mentioned at the beginning, in contrast to what all the other speakers, or most of the speakers before me have been talking about, I'm focusing on passive dendrites first, because we just like to start simple with a simple model of dendritic computation and see what that gives us. Right, so from a theoretical perspective again, what could dendritic computation give us? And this becomes clearer in contrast to the universal function approximators as we look at networks that have connectivity constraints. For example, consider this network where we have one neuron population that represents a scalar x1 and another neuron population that represents a scalar x2. Now, in theory, and these two target populations project into a common post population. Now, in theory, the functions that we can compute are of this form. So we have f1 of x1 plus f2 of x2, and we can also feed that into a neural nonlinearity. Now, no matter how we choose, no matter how we choose G or f1 or f2, we cannot compute certain functions in this network, including XOR or, more generally speaking, 4-quadrant multiplication. To actually obtain a universal function approximator that can do this, we need to introduce an intermediate neuron population that represents both x1 and x2, and then we can decode from the neural activities of this population a function f of z that could, for example, be the product between x1 and f2. But of course, the problem here is our goal, as I mentioned in the beginning, is to model neurobiological systems, and this might not be the connectivity that we find in the brain circuit that we'd like to model. For example, as far as I know, the models of attention in layers that make use of information in layer 6 of cortex do not really have neurons that represent both the attention signal and the signal that's being modulated. So that's the idea that's where dendritic computation comes in. The basic idea is that we now have a multivariate neural nonlinearity. For example, we can exploit the fact that we have excitatory and inhibitory input channels, and that these input channels interact nonlinearly within the dendrites. And this allows us to potentially compute a larger class of functions, but in contrast to this kind of network setup, this will not give us a universal function approximator. We just may be able to compute a larger class of functions. Now, to actually test this, we of course need a model of dendritic computation. As I said, we are using a very simple model of passive dendritic computation. In particular, what I'm calling here the n-compartment LIF neuron model. So the idea is we have one somatic compartment that's active and an arbitrary number of passive compartments and current or conductance-based input channels. So we can replace like these current-based input channels that we have by default in LIF neurons with conductance-based channels. And we can add like a single compartment that represents dendrites. So this would be a very, very coarse approximation of biology. We can like make this arbitrarily more complex. So here we'd have a 4-compartment neuron that has 6 different conductance-based input channels. Now, to actually integrate these neurons into NEF models, well, we need to do some math. So the first takeaway is that all these NLIF models, the sub-threshold dynamics, can be easily described in the form of a time-varying linear differential equation. Now, to actually integrate these neurons into the NEF, what we like to compute is a LIF equivalent current that essentially computes the current that's flowing into the soma given the input channel state G. And if we had such a function h of g, then we could solve a weight optimization problem that has the following form. So we're trying to find a weight matrix Wi that translates from the pre-activities into a average channel state that then results in the current that's desired according to our normative tuning constraint. And usually computing this function h is really hard because of the nonlinear dynamics of the somatic compartment. But the basic or crucial idea here is that as long as we have tonically firing neurons, then the somatic compartment essentially can be approximated as having a constant membrane potential. So we can clamp the membrane potential to this voltage V bar. And this should become a little clearer if we look at the voltage trace of a single LRF neuron that's tonically spiking. And the voltage just oscillates between the reset and the threshold potential. And if we analyze this in a little bit more detail, then we'll find that over a wide range of output rates and a relatively wide range of noise on the input side, we get more or less constant membrane potentials. So this assumption might make sense. And if we make this assumption, then solving for h is relatively simple. So now that we've solved for h, what on a theoretical level can we expect to compute within neurons of this form? So the first observation is that current-based and somatic input channels act linearly. So if we feed input into these channels, then the current that's flowing into the soma is essentially just a scaled sum of those input channels, depending on the synaptic weights. Second, conductance-based inputs can act divisively on other inputs of the same compartment. And lastly, and this is perhaps most interesting, conductance-based channels that are connected to a different compartment on the same branch interact multiplicatively with other compartments, with other input channels in different compartments on that branch. Now, again, on the theoretical level, let's analyze in how far, given this dendritic nonlinearity h, we can actually use this h to compute interesting functions. And kind of not to introduce any bias here, I'm generating these random functions of increasing complexity. So on the left, we are trying to approximate very simple two-dimensional functions. And on the right, we have more complex two-dimensional functions that we're trying to approximate. And on the y-axis, I'm plotting the approximation error if we're approximating these functions through one of the nonlinearities h that I've derived according to the model of our simple passive dendrites. So we can start this with a standard LRF neuron that just has a single active compartment, and then we get this kind of curve, which is relatively close to what we could accomplish if we just solved for functions of the form f1 of x1 plus f2 of x2. Now we can go to two-compartment neurons, and interestingly, we can compute a much larger class of functions using this kind of neuron. Particularly note the logarithmic scale on the error axis here. However, the errors that we get are slightly above what we would get with a multiplicative baseline where we have a function of this, where we are able to compute a function of this form. Now, the interesting thing is, as soon as we go to three-compartment LRF neurons, then we, for more complex functions at least, are able to outperform this multiplicative baseline. However, and this I think is also very interesting, if we go to the four-compartment LRF neuron that I showed you, then there's actually very little improvement over the three-compartment neuron model. Right, and also this is like the other interesting point here. If we were able in an intermediate population to generate a good two-dimensional basis, then this would be essentially the baseline, or what we could perfectly achieve in a network with an intermediate population. However, in practice, so when we implement all of this in an actual spiking neural network, things look a little different. So what I'm trying to do here is to compute four-quadrant multiplication. So we just essentially have a network where we have, in a pre-population, as we saw previously, we represent a scalar x1, and in another population, we represent a scalar x2. And I'm starting with this additive network, or essentially what we can compute with a simple LRF neuron. Over time, I'm varying x1 and x2 systematically, which gives us these spike rasters from the x1 and x2 population. And what we'd like to compute is a product between x1 and x2, which, as I said, is a generalization of XOR. If you can compute the multiplication between x1 and x2, then you're also able to compute XOR. Right, and for the one compartment LRF neuron, we're essentially getting a flat line as an output. It's not possible to approximate this function, as I mentioned at the beginning. Soon as we switch to two compartment neurons, we get this kind of output, which looks a little better, but we can also show in theory that this two compartment neuron that I depicted earlier cannot compute XOR. However, as soon as we go to three compartments, we are actually able to compute this multiplication function relatively well. And yeah, there's actually very little improvement if we switch to this more complex network that uses a comparable amount, but slightly more neurons than the network with the three compartment neurons. And again, note that these are very simple neurons that just use very simple conductance-based input channels and three passive compartments that are connected together, and such neurons can compute XOR. Right, we can also compare this for a large number of different benchmark functions that people might want to compute in their models. The interesting takeaway is that for most of these benchmark functions, the two compartment neuron actually outperforms our network that consists of two layers. The reason being that actually realizing a good two-dimensional function basis in this intermediate population, at least in a spiking neural network where we have to deal with a lot of noise, is really hard. Particularly note that the two compartment neuron is really good at computing this so the product between x1 and x2 over the positive numbers, which is similar to gain modulation. And yeah, as we just saw, if we go to three compartments, then we can approximate four compartment multiplication relatively well. So to conclude, we can systematically harness a very simple model of dendrites for computation in the neural engineering framework. Computation within these passive dendrites can sometimes outperform spiking multi-layer networks. Two compartment neurons can compute gain modulation relatively well, which might be interesting for people who are trying to model attention in cortex using these models. And three compartment LRF neurons can compute four-quadrant multiplication, or in other words, XOR. And adding more passive compartments is not necessarily beneficial due to a trade-off between the nonlinearity that we get with large coupling conductances, or low. So to get nice nonlinear functions, we need a small coupling conductance, but then distal input to our neuron barely affects the soma. And if we increase the coupling conductance, then we essentially just get linear interaction. And some of this, so in particular, the work on two compartment neurons was published in this neural computation paper earlier this year, in case someone is interested. With that, I'd like to thank you, and in particular my PhD supervisor, for working with me on this. All right, that's it.
Thank you.
Thanks for a very clear talk. So questions from the panel at this point? No one else yet. I had a question. You didn't mention that, and then I think Xiao-Jing is next, and then Bartlett. I had a quick question about inhibition. You didn't really mention inhibition. Is there no specific role? Because we heard a lot about inhibition being super important in many places.
So yeah, the model accounts for, so essentially in the neural models that I showed, actually let me bring up my slides again. In the neural models that I showed, I'm definitely including inhibition. So let's go back to this. So as you can see, each compartment is targeted by an inhibitory input channel and an excitatory input channel. And the inhibitory input channel and the excitatory input channel have like biologically plausible reversal potentials. And just the weight solver cares about how inhibition, how excitation and inhibition are actually combined.
Okay, then Xiao-Jing, you have your hands up.
Yeah, I have a very quick technical question, which is when you reset after spike, what do you do? Do you do something special about dendritic compartment? Another question is, please explain again, how do you get a multiplicative gain with two compartments?
Right. So to answer your first question, I'm not doing anything special. So I'm, in contrast to, so I am actually, did I have a diagram of this here? Right. So I am actually including a simulated spike that backpropagates through the dendrites whenever a spike is generated. And I'm not resetting the dendritic compartments in any way. So there's backpropagation of voltage of the spike potential to the dendrites. And to answer your second question, maybe I went over this too fast. In the two compartment model, so this model, you can't get multiplicative interaction between the channels. So according to these rules that I have here, you only get multiplicative interaction between input channels that target different compartments. And in the two compartment model, there was essentially only one compartment that was not direct, that was separate from the soma. So you only have this divisive interaction between the two input channels in the two compartment model. But in the three compartment model, essentially ignore this branch up here. In the three compartment model, you actually get multiplicative interaction between the conductance based inputs, and that just falls out of the math after you've derived this multivariate nonlinearity function H.
Thank you.
Bartlett. Oh, you muted.
Yeah, no, thank you. I got it. Thanks. Andreas, the question I was asking, I'm trying to figure out what kind of level of abstraction that you're working at here. And I think I didn't quite catch that, but I think it's essentially a mathematical model. When you're talking about compartments, you're not simulating a neuron with, kind of in a more biophysical setting. And I was wondering how many of these conclusions would hold up under, for example, the idea of being able to compute these different kinds of functions and whether you're successful or not successful. If you were to move to a more biophysical kind of modeling level of abstraction where you actually, it's not so clear what a compartment is. When you have a neuron, you have dendrites. I mean, there are cables and things are, there's branching and there's other stuff that doesn't lend itself to these very simple mathematical abstractions. And I was kind of wondering if you think that the kind of conclusions, these sort of firm conclusions that you're coming to about the computing capabilities of different numbers of compartments would survive moving to, a more cable-like model with AMPA and NMDA and GABA and, all this other kind of stuff.
Yeah, I think, so that's a very good point. Like I'm not claiming that this is extremely biophysically detailed. So again, the goal here was to start simple, to incorporate like a biologically inspired model. You might even say it's like biologically plausible into our networks that models dendritic computation on a lower level and thinking about what we can potentially do with this. Regarding the question of which results would hold up. So I think there's trade-off between coupling conductances and nonlinearity is something that you could also apply to more biologically detailed models. So again, the coupling conductance would essentially correspond, so the strength of the coupling between the compartments essentially corresponds to the distance between the compartments and the thickness of the dendrite at this point. So I hope that there might be some way to very abstractly map more detailed models onto these kind of very simple models that do not necessarily use cable equations.
Yeah, I think that would be interesting to see. Thank you.
Right, thanks. With that, let's give applause to Andreas and move on to our final presentation. Last but not least, Xiao-Jing Wang will tell us more about network computations, I think. You're muted. Okay.
Here we go. Thank you. So the theme today for my talk is gating, and I'm going to talk about dendritic disinhibition as a mechanism for gating. We became interested in gating when we were exploring circuit mechanism computationally of working memory, which is our ability to hold internally some piece of information in the absence of external stimuli. As you know, in order to be able to hold information in your working memory, your brain needs to have the ability to filter out all this incoming information flow that's not relevant behaviorally. So for that purpose, we wondered whether we should, consider specific gating mechanism for inputs onto dendrites. We were impressed by the anatomical observation that different kinds of interneurons have different proportionality in the prefrontal cortex, which is known to be important for working memory relative to primary sensory areas, where PV expressing interneurons are predominant, which is not the case in the PFC. So based on some anatomical evidence, we developed a local circuit model for working memory that incorporates three kinds of interneurons. In addition to the PV expressing interneurons that controls the spiking output of pyramidal cells, you have a second type of interneurons that may express somatostatin or carbendin that target dendrites, with a third type of interneurons that may be labeled by VIP or carotene in green, preferentially target dendrite targeting interneurons in blue. So you have, depends on the activity in blue or green, you can either close the gate when you have too much dendritic inhibition, or open the gate when the blue cells are suppressed by the green cells. Okay, so that model was proposed theoretically in 2004. So here's a simulation where you see the activity of pyramidal cells along the y-axis, say tuning for a particular direction as a stimulus feature as a function of time, right? So you see that the brief input can trigger a tuned activity pattern that's self-sustained across a delay period. But you also notice that for those neurons that are not engaged in coding the behaviorally relevant stimulus are actually suppressed compared to baseline activity. And so you can look at the tuning of different cell types. Of course, the model would predict that for SST neurons, you'd see inverted tuning, right? So you'd see that for some preferred cue, you have a reduction of activity relative to baseline. And we look at single neuron recording data from behaving monkeys and found indeed there is a small fraction of neurons about 5% of recording neurons that show this inverted tuning relative to the baseline before the cue is presented in a working memory task. We also explored how dendritic inhibition might really help to kind of screen out distractors by changing the relative ratio of dendritic versus somatic inhibition, and found indeed that while the inhibition in total is preserved, you have more dendritic inhibition as you would expect. Your working memory representation would be more robust against the distractor than the other way around. Okay, so now we think that maybe this kind of gating mechanism is more general than just for working memory representation. Of course, now we know that this kind of disinhibited motif is commonly present in all cortical areas, including sensory areas as well as the prefrontal cortex. So when we start to think about multi-regional cortical system, I think there's really a number of very interesting new questions we have to confront. One of them would be gating, all right, routing of information to the right place at the right time. Given that connectome tells us that there's a rather intense, the connections between cortical areas are rather dense. Each area gets input from several dozens of other cortical areas. There must be a way to route the information to the right place at the right time, depend on the behavior demands. Very simple example I want to mention is sensory information from the auditory and visual pathways, right? So this is what we call pathway gating, right? Normally, like now, you integrate visual and auditory information in some higher areas, but you don't necessarily always want to do that, right? So for example, if you're reading a book in a noisy cafe, you may want to get in visual information so you can read a book, but get out auditory information so you don't hear noise around you, right? So the question is whether this kind of disinhibited motif can also be applied for this kind of routing of information flexibly. Robert Young, when he was a graduate student, explored that possibility in a model where you have pyramidal cells that have a number of dendritic branches that are more or less independent. And imagine that you have visual inputs and auditory inputs that are somewhat clustered. Okay, they don't really completely overlap. You also imagine that there is this disinhibited motif. Now you have several of them, which are somehow targeting different dendritic branches, right? And then you can imagine, what's important is this nonlinear dendritic responses. So Robert Young actually went a certain distance, certain length to capture this plateau potential in the model that's very similar to what's observed experimentally. That turns out to be important because if it's just a linear response, the gating will not work as well, right? So imagine that you have pathway 1 and pathway 2. When the inputs from pathway 1 are disinhibited, indicated by dashed line, but not pathway 2, then you get in the inputs from pathway 1, but not pathway 2, right? By contrast, if both are closed, you will not see any difference for the inputs from each of the two pathways, right? And you can see that by opening the gate for pathway 1 but not pathway 2, now your response becomes rather selective for only pathway 1. All right. You say, okay, this is kind of made up a structured model. Does it still work if you are using a more realistic random projections? So we went through some kind of network analysis, assuming completely random projections with the numbers we could find in the literature. We found that the gating is pretty good, insensitive to most of those parameters, except the number of somatic, SST neurons that target a particular dendritic branch, right? So you see here, if that number becomes too small, then you lose the gating capability. We also wondered how this alignment of disinhibition and activation from a particular pathway can occur dynamically. And so we used a learning rule from Grupner and Brunel, taking into consideration that the learning curve as a function of the firing rate depends on inhibition, right? You can imagine that suppose that initially you don't have this alignment. So pathway input goes to all branches, but you just open the gate for one of the branches, right? So in that event, because this dendrite is disinhibited, you are kind of in this situation, so you can have LTP. But in the other branches, because of the inhibition, you are, say, in the other situation, you have LTD. So through plasticity, you can imagine that this alignment can occur, dynamically, and that's shown, confirmed here, right? So before learning, you don't have selectivity, whereas after learning, you get this gating, working. Another very important finding, I think, when we move into large-scale system, is the observation that if you look at the proportion of interneurons that are somatostatin positive versus PV positive, so input controlling versus output controlling, it turns out that that ratio is different from area to area, and high areas, association areas, including prefrontal areas, show higher input controlling versus output controlling neuron ratio than early sensory areas, as you should expect, right? For higher areas, you need more input control than early sensory areas. So this is what we call macroscopical gradient of inhibition. So now, for the last part, I want to talk about a new story where we try to build a large-scale model with many interactive cortical areas for distributed working memory representation. This is summarized in a very nice review showing basically the literature as a kind of meta-analysis about in the macaque cortex, which areas in the cortical system of the macaque tend to show delay activity in red, which areas tend not to show delay activity in blue or absent of data. So working memory representation is not localized in one particular brain region, and we want to understand how that works. So this is a kind of a last version of our large scale modeling, spearheaded by Xiang in collaboration with Nicola and Carl Zillers. So this incorporates the model, namely incorporates different kinds of data. On one hand, Nicola and Carl measured receptor labeling density using autoradiography. In particular, I'm going to talk about the dopamine D1 receptor labeling in macaque monkey cortex, which has been known to be important for gating and for working memory storage. We also used a new set of inter-area connectivity matrix measured from Henry Kennedy's data laboratory. And the measured density labeling data should be normalized by neuron density. So we can have a better capture of dopamine modulation per neuron. And the neuron density actually varies from area to area, as you know. And that's coming from John Cass's lab. And finally, we used a spine count per neuron from Guy Elston as a proxy for the scanning of the strands of excitation per neuron. And that also changes from area to area. It's quite different, say, for a neuron in PFC than in an early sensory area. So we have to combine those data, namely Xiang combine those data in a common cortical space. And then I guess the new experimental finding is really from the receptor density labeling, which quite nicely show, as you see on the right, this macroscopical gradient as function of the hierarchical position defined by anatomical method for different cortical areas, with V1 being at the bottom and frontal parietal areas higher in the hierarchy. All right, so that demonstrates that dopamine modulation is not the same across the cortical tissue. It's also not just randomly heterogeneous. It shows this very simple low-dimensional gradient across cortical mantle. All right. So the question is, how do you incorporate dopamine modulation in a functioning large-scale system? By the way, the connectivity, I said, so I don't show the data, is coming from Henry Kennedy's lab. We were very impressed by an early study from Pat Goldman-Rakish's lab. This is a rat slice work showing basically back then that dopamine applied in the bath enhances dendritic inhibition. The recording was done from soma, so you see a small change from white to red, but you see an enhancement of dendritic inhibition, and you see at the same time a reduction of somatic inhibition, right, by dopamine. And that's very interesting, given what I told you earlier, that perhaps dopamine acts in multiple on multiple targets, but one way is to selectively enhance dendritic inhibition versus somatic inhibition, right? So we incorporated some other experimental observations, for example, from German Siemens, showing that dopamine D1 enhances NMDA-mediated excitation, as well as GABA-A-mediated inhibition. And Amy Anston has some evidence that if dopamine modulation is too strong, you activate some potassium channels, which in fact reduces excitability of single neurons, leading to the so-called inverted U-shape dependence of dopamine modulation of working memory. So we incorporate all this in this large-scale system. For that purpose, each local circuit now also includes this different kinds of interneurons, right? SST expressing, PV expressing interneurons, as well as VIP interneurons. So you have this large-scale multi-regional model really across levels, from different cell types, different ion channels, all the way to multi-regional large-scale cortex. So this is one simulation where you see the activity map during the stimulus presentation, followed by a delay period. Right, so you see now during the stimulus presentation, the activity is mostly confined in the posterior part of the cortex, but during the delay period, when the input is gone, the persistent activity that encodes working memory is mostly in the frontal areas, right, anterior part of the cortex. By the way, we have stimulus selectivity in this simple simulation. Let's say you have only two stimuli, red versus blue on the right versus left. Okay, and you see that the response in V1 is transient, only during the stimulus presentation, but a frontal area like 46D shows delayed activity that's self-sustained during the delay period. And that's when the dopamine modulation level is a parameter that scales the gradient, right, which is the spatial structure of dopamine modulation. Maybe I should have said this one more time, that the dopamine level would act on those different targets parametrically like this. right? So this is one dopamine level is at a certain level, which is optimal. But if it's lower or higher, if it's lower, you reduce recurrent connections, you don't have enough reverberation, so the persistent activity is absent. And when your dopamine is too strong, as I said, the neuronal excitability is reduced, you also don't have persistent activity, right? So that leads to this inverted U-shaped dependence of the dopamine modulation, except now you can show that selectivity for different cortical areas in a large-scale system. Now, quite to my surprise, the model actually captures essentially the collected data from more than 90 experimental studies with macaque monkey summarized in the plot I just showed you. All right, so you see the just visually that the experimentally measured connectivity pattern, persistent activity pattern is very similar to what you see in the model. Now, there is an interesting debate recently about whether certain form of short-term memory trace can be realized without persistent activity. And that can be in a hidden state, for example, by virtue of short-term plasticity of synapses. We incorporated short-term plasticity into this large-scale system, and we found that indeed, sometimes in this case, you can, encode a particular stimulus in red, for example, in the form of synapses, and you don't see persistent activity during the delay period. The problem is that when you want to introduce a distractor after a stimulus that's relevant behaviorally, the synaptic strength would always be the biggest for the distractor, which happened later, right? Because the synaptic plasticity trace decays away with time, so it has decayed less for the blue distractor than for the red relevant stimulus, right? So as a result, this activity silent state scenario is okay if you don't have distractors, but it's not okay when you are confronted with the issue of filtering out the distractors. It's also not okay, by the way, if you need to manipulate the information during the delay, right? And working memory includes, by definition, the need for manipulating information internally in the absence of external stimulus. So activity silent state scenario, it's fine for some passive short-term memory traces, but not really suitable for working memory representation for the reasons I just gave you. And finally, we found that actually dopamine modulation can switch the system between the two regimes. So if it's not enough, you have this activity silent scenario, but if you increase the dopamine modulation, you can switch easily from the passive activity silent scenario to the active persistent activity mode for short-term memory operation. So let me summarize. So the focus again is about gating. This disinhibited motif, which is widely known today, was actually initially proposed theoretically, and we propose provides a potential mechanism for what we call pathway gating. This raises a number of interesting new questions, right? So you can ask whether you do have multiple disinhibited motifs that are somehow dedicated to gating different pathways, right? So that's still open question. And secondly, this macroscopic gradient of input controlling versus output controlling interneurons is very interesting, I think, and that is valid both for rodents and for macaque monkeys as well as humans. We start to incorporate that as well as dopamine D1 receptor, modulation gradient, which in part, as I showed, acts through selectively enhancing dendritic inhibition while reducing somatic inhibition. And that may be really important for filtering out distractors, as well as potentially switching between this activity silent mode versus active activity mode for short-term memory trace, which is passive versus active working memory. Thank you.
Thank you.
Thank you very much. We have a few minutes for questions for Xiao-Jing before we move on to the panel discussion that Michael will guide. I had a question which has to do, a lot of folks are studying oscillations in the brain, and they think sort of that maybe gamma oscillations or other oscillations set up functional dynamics on connectivity and networks. Do you not see a use or need for that? Is that another way of thinking about it, or is it could one combine those two ideas? What do you think about that?
Yeah, so we did have a paper about oscillations in a multi-regional large-scale cortical model, and I was focusing on feed-forward versus feedback projections, right? So when you have this, large-scale cortical system endowed with a hierarchical structure, it's really a good platform to look at how feed-forward versus feedback, top-down versus bottom-up processes interact. And there's evidence that feed-forward processing somehow selectively enhance gamma oscillations, whereas feedback projections potentially coming from deep layers in source areas, enhancing either beta rhythm or gamma rhythm. So we have a version of the large-scale cortex model with a laminar structure in each local system. We did not really have dendrites, so I did not talk about it for this particular workshop. Potentially, it's worth exploring in the future if there's strong evidence, for example, that beta somehow involves somatostatin interneurons rather than PV neurons. It's quite well established that gamma arises mostly in superficial layers through interactions between excitatory neurons and PV interneurons. But the second mechanism for beta or alpha, there are multiple proposals, but less established.
Thank you. And I think, Bartlett, you have your hand up?
Yeah. XJ, thank you. I was very interested in thinking about what you talked about. You have this picture that recurs in your slides, and which shows pathway 1 coming in, and there's a disinhibition or inhibition of dendrite 1 and pathway 2 and dendrite 2 and so on. And it assumes that the disinhibition is very articulated. So it's like per dendrite. I mean, I don't know if you mean that literally, but that's kind of what my question is. Do you think that the somatostatin system, which then can be disinhibited, or I should say inhibited and thus disinhibiting the neurons, is kind of operating at the level of selecting individual dendrites of pyramidal neurons in the cortex, or what? Because I don't know of evidence that individual inhibitory neurons like target just this one dendrite of a neuron and become that dendrite's private inhibitor or disinhibitor. And I'm kind of wondering what level of granularity do you think that system has?
Yeah, so that model definitely assumes some clustering, but I maybe went a bit fast. One slide where we actually assessed whether this is still possible if your projections from somatostatin positive neurons to dendrites were completely random. So you don't have this, branch to branch specificity, right? And the answer, the conclusion is yes, as long as you don't have too much feedback. So that's, and that's something that experimentally, I guess, can be tested, or maybe more computational model would also help. But in our hands, with the numbers we found in the literature about the number of somatostatin interneurons, the probability projections within a column, you can still have it even if the projections are 100% random. There is also another assumption, right? That the pathway specific excitation is somewhat clustered. That's also an important assumption. There's a bit of evidence from Wenbiao Gang actually about that, but and frankly, this kind of data can now be collected experimentally if someone is willing to do it.
Thank you.
Thank you. I think because in the interest of time, we should move on to the panel discussion where we're trying to tie together various messages and distill what we can learn from each other. And Mike Hosa is taking on that interesting job.
Yeah, hi, everyone. I just wanted to start by adding my thanks to all the speakers for their really fantastic talks. And thanks also to Dieter for organizing the workshop. I thought I'd open the discussion with the title of this workshop, which is, Are Dendrites Necessary for Cortical Computation? And I'm kind of assuming that all of the speakers here would answer yes to that statement. Any of them want to challenge it? Maybe Brent?
Sure, I'll play bad guy. I'll play bad cop. Sure.
All right.
So I used to do, I'm an ex-dendrite pat. In my PhD, I did a lot of work in dendritic dynamics in terms of bursting mechanisms. And then I sort of moved on from there, sort of getting more excited about rich circuit structure. And I guess throughout my career, I've been hesitant of including dendritic modeling, sort of more going towards a little bit simpler reductionist frameworks and trying to ask and answer questions that live there. And I seem to continue to move forward on this. So so I don't need to appeal to dendrites to sort of push my science forward. Is this a mistake? I'm not sure, but I don't feel the need to do it. I'm not being pushed necessarily. I think what will happen though is with the events of voltage sensitive dyes, if we start to get more information about the spatial temporal structure in experiments, then maybe I'll be forced to do this. Anyway, I'll put that out there. I haven't had the need to do this. Am I missing out? Probably I am, but I'll throw it up. I'll throw it back to anybody else.
Okay. I can give you my perspective, which is that, I think there's a lot of evidence, as you've seen from the talks today, that the dendrites can perform valuable computations, but there's still a gap that needs to be crossed if we're going to show that dendrites are actually necessary for cortical computation, as the title of the workshop states. I think that that's going to require not just observation of dendritic dynamics during behavior, as Brent has just suggested, would be possible with voltage indicators, but actually some kind of a deletion experiment to delete a dendritic mechanism, whether it be a specific nonlinear channel, for example. If you can do that and show that this actually kills the computation and thereby affects the behavior, this would be a really crucial experiment. Alternatively, if you can show that dendrites are performing a computation that is unique and that can't actually be performed by a circuit, that would also be a really important demonstration. And I'm not sure we've identified such a computation yet. Yiota, I mean, you showed us this incredible catalog of different logical operations that a simple dendritic mechanism can achieve. But actually, all of those mechanisms can also be done by networks, right? These are not uniquely dendritic computations, I think.
Absolutely. And I personally don't think that dendrites are necessary. I mean, they may be necessary in our own scalp or within an animal scalp because of pressure of our brains to evolve under a limited size or, limited energy requirements. So they became necessary out of necessity, let's say. But if we had the freedom to choose a very large, network of point neurons, and we had enough size and energy to run with it, So far, I have not found the type of computation that would not be solved. And I'm very disappointed to say that, but I haven't been able so far to find something that you wouldn't be able to do without dendrites if you did not have these constraints, right? And I'm not sure if there's a way to test this, because all of the animals that we are studying, they have evolved under these constraints. So how do we actually test it?
Arnd, you had a comment?
Right.
Or Bartlett?
I do, yeah. I think it's important to kind of separate that question. Not are dendrites necessary or not necessary. I mean, there's this biological question. If you're a scientist and you look under the microscope and you're looking at neural tissue and you want to know how it works, and let's say it turns out that dendrites are separate units, and a dendrite fires and it could learn. And you used to think that, oh, only the neuron could do that. And now you've learned that a dendrite could act like a neuron or something like that. It's not that dendrite that you're now, or that functionality that you're now attributing to a dendrite is a familiar functionality. It's something that, like you said, if someone says, oh, it's like an AND gate, well, I already knew how to do an AND gate, so I didn't learn anything new. Now, the biologists learned something new, is they're figuring out how the brain works and how the circuit works and how the integration occurs in the dendritic tree of one neuron. And that would be important information to know if you're trying to figure out how the brain works. And obviously they're necessary to be there for the brain to work, but it's not a new type of weird computation. Now, but I want to add, you said, are there examples of weird new types of computation? And I apologize for referring to some of our own work. So the one thing that we've not so recently, maybe in the last 10 years, found that was different from what a conventional summing unit or a conventional logic gate would do had to do with spatial interactions among synapses on dendrites, where, for example, the interaction between synapses that target more proximal or more distal parts of a dendrite is an asymmetric nonlinearity that is unfamiliar. It's not a kind of an operation that you're used to thinking about. And we found that it was well suited to do kind of classical contextual interactions. So there were it was a device that took advantage of the cable, the extended cable, and had nonlinearities and NMDA channels and things, and was a different kind of device. It really was a different kind of device. Now, is it used? We don't know yet. But because we still don't know where synapses actually target on dendrites, to a significant degree, we don't know a lot still. But there are examples because of the biophysics and space that are devices that are not familiar and are new types of things. And we don't know yet how much that is relied upon, but it is different.
Great. XJ, you had a comment?
Yeah, I guess as you can see from my talk, my take is perhaps on one hand, we all think there's a canonical circuit organization, right, that applies to all cortical areas. But it's also, I think, useful and important to realize that there are differences across different kinds of cortical areas, like Bartlett Mel also showed for the spine profile in sensory versus frontal areas. I also showed this macroscopic gradient. So in higher areas, you have a lot more dendrite targeting in the neurons. Maybe one more piece of data which just came out, which really I think is pretty shocking, right? We tend to think that EI ratio is something like 4:1 or 5:1, and that's indeed the case in mice, but that's not the case in primates. So in human, the EI ratio is 2:1. You have a lot more inhibited neurons, and in association areas, I bet you have a lot more dendrite targeting interneurons. So I guess it's an experimental question and computational question, right? How we can really bridge species and bridge, what we learned, say, from sensory areas with what might happen in higher association areas.
Cool. Thanks, XJ. Arnd, you had a comment?
Yeah, thank you. I have a comment going back to Brent's initial statement, where I would say that dendrites definitely are a complication that one may want to avoid, but they also confer benefits that may explain things that we don't understand how the brain does it. For example, when you think of how deep networks learn, there's this backpropagation algorithm that's nothing to do at the first glance with exponential backpropagation I was talking about. But how biological neural networks can implement that is really not solved. And it may very well be that dendrites very much are the substrate on which this is being solved by separating in space the thresholding for the ongoing processing, the spiking of every neuron while we think and act. And at the same time, we don't have to phase this in and out between different cycles. At the same time, we can learn using dendritic mechanisms that are can occur concurrently with the ongoing processing. And so that is something, of course, that artificial neural networks don't need to do because you can train them separately from their use, but animals can't usually do that.
Good point. Would any of the other speakers like to weigh in?
I would, if I may.
Go for it.
I fully agree with what Arnd just said. I mean, by looking at how the brain is doing it and how dendrites contribute to different kinds of learning, then I think we learn a lot of valuable lessons that can then help us not only understand what it is, a black box like a deep neural network, let's say right now, but also devise different learning rules that maybe try to mimic the brain, because the brain is doing it like this for reasons of efficiency. So I think there are very valuable lessons to be learned there. And for example, one of the very recent things that seem to come out in the machine learning community is that dendrites seem to help by increasing sparsity, let's say, or by helping a system continuously learn information without forgetting the previous ones, because it's a very efficient way of building modular networks, so distributing the information to different parts of the networks because of the tree-like structure and the constraints that come from the connectivity of dendrites. So that, I think, it's a very valuable thing that we can learn by studying the dendritic structure and function and the way the brain is doing it to devise better, smarter systems.
Great, thanks, Yiota. Maybe I can throw in a question from the audience. This is a question from Alexandre Gouet-McCrate, who's asking, in circuit modeling, is hesitancy to model dendrites more so due to technical computational limitations, the speed of the simulations, or a point of view about the importance of their function? Would anyone like to pick this one up? Bartlett, Brent, XJ?
Well, people in deciding what to include in a model need to kind of decide what they're trying to model. I mean, it's like, it's not such a question of, well, should we throw in dendrites into a model? I mean, there better be a reason to put anything into any model. It depends on the question that's being asked and so on. And I can imagine all kinds of reasons for hesitancy to include things that people don't understand or why they would need it. So I can't give a very satisfying answer to that question. I personally think that when the question about circuit level modeling, I think that anything interesting or different that dendrites of real neurons do compared to the units that we're used to in OR gates and summing units and ReLUs and things like that is going to have to do with the architecture of the neuron. You have apical dendrites and you have some kind of amplification up there. You have within dendrite interactions possibly. There's even evidence at Mark Harnett's lab. They have, they were showing that different dendrites have different levels of NMDA channels, sort of sensitivity. And so you have sort of a tailored way of taking advantage of different dendrites, maybe with different levels of nonlinearity. And so then when you put the circuit around it and you have XJ has all these pictures with different kinds of inhibitory neurons that inhibit and disinhibit and target dendrites in different ways. I mean, now you're talking about a pretty complicated machine. And nobody really, we don't yet understand the kind of expressive capabilities of that machine yet. I mean, people are exploring, like XJ showed, different things that you can do. And then of course you can modulate with dopamine. I mean, there's a lot of knobs that can be turned in this circuit. And I think maybe focusing on dendrites in isolation from all the circuit in which it sits with all these other things coming into it with modulators and specific targeting is really the interesting kind of meat of the problem. But there's no pithy single answer to it. And, oh yeah, I'm going to throw it in. So we have a ways to go before we can kind of pull that whole thing apart and sort out what all the capabilities are, I think.
I think also I often am pragmatic in the sense that when I start building a model, it's often in collaboration with an external team, and I want to make predictions for them to test. And I know if I'm going to go down the dendrite exquisite complexity dendritic route, there are very few people I can collaborate with that can really test in a serious way these kinds of predictions. I think, you know, the world we live in now is, you know, large scale data sets of, you know, single cell image neurons that are relatively simple. I do think that with the voltage indicators, if that comes to bear, it'll be a new renaissance for dendritic computation, much like it was 20 years ago when sort of the slice experiment world sort of pushed us that way. But it was tough to be a theorist in that world. I mean, Bartlett and other people here have done really well with that. But if you're really just giving me two spatial points on that dendritic tree, the soma and the dendrite, and maybe if you're world-class patcher, you get another one, right? Like somebody like, you know, Michael here. But I don't know. There's only so many things I can really, you know, put forth that. But if you're going to give me the whole spatial temporal distribution in a movie, well, it's a game changer. So the world may change a lot when we get better data, and then I think we'll see. I think it'll be a renaissance. It's exciting.
Yeah, I totally agree that a voltage sensor that can be used to image dendrites in vivo would be a real game changer for the field. Unfortunately, we're running short on time, so I'm going to throw this back to Dieter, our chairman.
Right. Thanks, Michael, for a wonderful discussion. I think that really brought some of the very important topics to the forefront. And just as a very final comment, obviously the human brain and its cognitive abilities are still far beyond anything that any artificial network does. So there is room to learn a lot of new things in the next generation. I'm looking forward to those. And I'm definitely looking forward to the voltage imaging, which we're also doing in our own lab, but mostly with large scale, non-single cell resolution. So there are also multiple ways of doing that. So thank everyone for their great contributions, for a challenging workshop on challenges for us in the future and challenges to relate to all the different things to each other and see how they could add up to cognition. And yes, it will keep us busy. And again, hopefully we see each other at various meetings in real life with real persons, and also the folks from the YouTube and have a coffee and drinks and all that good stuff as we move to the post-COVID world, even if COVID still remains. So thanks a lot and goodbye.
Thank you, Dieter.
Thank you, Dieter. Thank you, everyone.
Thanks, everyone. See you.
Bye.